Hello, everyone, and welcome to our last talk of the second day of Varsity Sci. Well, last session. Today we have two talks lined up. Both will be on plant sciences, which, as everyone knows, is a brilliant subject, and I'm not biased at all because I did plant sciences in second year. So our first talk will be from Christian Norton, which will be on Arabidopsis genotypes and their fitness differences, whether they show fitness differences depending on soil community. Our second talk will be from someone who will be very familiar to people who are following BioSoc talks, from Ella Cafferel, and she'll be talking about the deep, dark secret of the algal chloroplasts. I wonder what that could be. Without any further ado, I'll pass on to our first speaker, Christian.
Hello, everyone. Can you hear me fine? Yes? Okay. My name is Christian Norton, and I'm a DPhil student at Oxford in the Department of Plant Sciences, of course. And I'm in the Hector Turnbull lab group. And these online presentations are kind of fun because I actually have no idea if anyone's watching besides the admin people who are the organizers. And also a thank you to the organizers for doing this. It's actually been really amazing to hear across a wide range of topics that I generally wouldn't hear about. I often don't hear about swans or viruses or other things, so it's been really enjoyable. So this is the name of my talk. Do Arabidopsis genotypes show fitness differences among communities? So that's the official name of this research, but often I have another title when I'm giving talks, and because as a scientist, as a science student and as a scientist, I often find that it's hard to explain what I do to my family. And so this title is a joke. There's no frustration in there. But just to kind of clarify what I do to my family, I often will tell them that I'm just a gardener. But I find that the byline is actually really helpful, though, because it is actually a story about plant roots and the tiny critters being very general, that live in the soil and how they interact. Okay, so I think one of the drawbacks from these online conferences is that we don't really get to meet each other or interact. So I put the slide in here. That's my face. I'm Canadian. I did my undergraduate in Canada. I did my master's also in Canada in Montreal, and now I'm at Oxford with Lindsay Turnbull doing an ecology PhD. And just to step back a second, my undergraduate I mainly did developmental floral morphology, so using SEM, scanning electron microscopy to image developing flowers. And then my master's was a lot of, it was cultural botany. And so I was learning about plant use in the Canadian subarctic by Inuit communities. So I've really been like all over the place in terms of the remit of what plant sciences covers, from developmental stuff to more social science areas, and now I'm into the ecology side of it. And so this is a very simple diagram that attempts to explain what I assume is like an infinitely complicated remit of relationships between plant roots and soil microbes. So we know plants have roots. Generally, roots are in the soil, and this soil is full of microbes. The microbes can be oomycetes, bacteria, I suppose viruses. And so the roots are interacting with this microbe community in the soil. And we know that plants have an ability to regulate or influence what microbes live in, on, and near their roots. And we also know that microbes have consequences for plant fitness. Some microbes are beneficial, some are harmful, some are quite neutral. There really is a whole range of ecological interactions between these plant roots and these microbes. So you have these two pieces, the plant roots and the microbes, and we know this background information about plants moderating the root microbiomes and the consequences of certain microbes. And so it leads us to this big question, and that is, what role does the soil community play in shaping the above ground plant community? We could easily go the other way with that and ask, what role does the plant community play in shaping the below ground community? But I'm interested in how the below ground could shape the above ground. And that's a very big question. We kind of get at that question by having these two more testable questions. And so moving to Arabidopsis, do Arabidopsis genotypes show fitness differences among soil communities? And further to that, do these genotypes differ from each other in their responses to different soil communities? Okay, so this is the system that we've set up. I have my 10 Arabidopsis genotypes and my 4 soils from Whiteham Woods. And so let's step more into the system now. So for the genotypes, the 10 genotypes, these genotypes span the chemotype diversity for Arabidopsis. And so Arabidopsis, they have these defense compounds that are known as aliphatic glucosinolates. And they can take on basically 6 different forms. And these 10 genotypes that we picked, each of these have, they span the variety of chemotypes for Arabidopsis. And the differences are based upon the carbon chain of the glucosinolate and also the functional group that's attached to the glucosinolate. And then we have our soils from Whiteham Woods. So Whiteham Woods, I believe Chris Perrins might have mentioned Whiteham Woods during the keynote, but it's this large research woods that's associated with the University of Oxford. And this is where I chose to collect my soils. And so I chose 4 soils from Whiteham Woods, the reedy fen and the semi-natural ancient woodland are those 2 red dots on the right. And then the ex-arable field and the grassland are these 2 dots on the left. And the reason we chose these 4 sites was because we know that one of the main predictors of differences in soil communities is the difference in the above ground plant community. And these four sites do have different plant communities, and they also have different life histories. The reedy fen, the semi-natural ancient woodland, the ex-arable field, which obviously would have been agriculture with crops in it. And then we also have the grassland, which has been, I believe, chalk grassland since the 80s. So we have these. So these sites were picked because we went in with the understanding that they should have different soil communities. Let's say that. So this is the setup for this experiment. This is 1 plant per pot, and obviously their positions on this bench in the greenhouse of plant sciences was randomized. And so we have each genotype grown on each soil plus a control with replication. And the plants aren't grown on all on one soil type. They're grown on an inoculum. So I have sand and vermiculite, which is obviously autoclaved. And then I mixed in 10% of bulk soil by volume. And then the control is simply a mix of all 4 soils that is then autoclaved. And then to account for chemical differences and nutrient differences, we added a nutrient solution to this. And so this is what the bench looked like in plant sciences. And each of these pots contains one plant. What did we find? And so we ran a 2-way ANOVA with interaction for both the width and the dry mass, because that was the data we collected from these plants. Measured the width and then dried them in the oven and took the dry mass measurement. Dry mass and width, the results and the trends were almost identical. So I'm just going to focus on the width just for time's sake. And if we look at the results, we found significant differences among genotypes and among soils, meaning that genotypes had different widths, which isn't particularly interesting to know that, because going into that, we knew the genotypes had slightly different morphologies. So that's not particularly interesting. It's great to see that there was a difference among soils, meaning that the width depended on what soil it grew on. And then even more importantly, there was a significant interaction between genotype and soil. And so I'm not sure kind of like what background people have in terms of stats. And so when we see a significant interaction, that tells us is that the trends we saw for genotype width and for width among the soils, this combination of genotype and soil produces this new effect, this new trend that's different from the genotype trend and the soil trend alone, which tells us something about how these genotypes interact with these soils and how that's somewhat unique to the genotypes. And so if we look at that just at soil first, we do see that the control is the first one on the left. The plants did best on control, and then X arable is obviously X, fen, grassland, and woods. And we see a general upward trend from X arable to woodland for the width measurement. And so obviously plants that are bigger or wider have more biomass, and we could probably say they're more fit. And so this is the general trend for soil. But if we note that interaction was significant, we can look at how that changes across the soil types as per the genotypes. And we know that interaction is significant. So Columbia, the one on the left there, COL, its trend is very much like the general trend when we considered all the genotypes together. But in the case of the MA genotype, Ma, we can see that the fen and the grassland, instead of being about equal as per the main trend, the fen, the MA, the Ma did much better on the fen soil. So now we're starting to see that these genotypes express these individual patterns in terms of what soil inoculum they showed a better or worse fitness on. Looking at dye, the dye genotype and the node genotype, dye had an incredible variance in the control, but then the other 4 soils showed a similar trend to the main trend. But then in the case of no, we see such a range, both in the control and also within the fen and the grassland. And so now we can start to see that we do note these fitness differences amongst soil types. And then further to that, we note that these genotypes have fitness patterns among these soil types that are unique to those genotypes, and that is confirmed by this significant interaction from the ANOVA. And this interaction plot really clarifies things for me, because if there were no interaction, it's not not the scale bar, the legend isn't shown because I could get it to plot right with R. But each of these lines on the plot is one of the genotypes. And if there were no interaction, there would be no lines crossing on this plot for the mean width. But because these lines are crossing, that shows us that these genotypes are having responses among the soil types that are unique to that genotype. And so we are seeing that interaction. And so how do we move forward now that we can comfortably say that yes, there likely are these fitness differences. Back to the questions, I should clarify first. Do Arabidopsis genotypes show fitness differences among soil communities? And do these genotypes differ from each other in these patterns? So yes, there was the main trends where they grew best on the control, which was the cooked soil, the autoclaved, and then improving from X-arable up to grassland. But then that bottom point, yes, the genotype showed individual patterns concerning how well they grew on which soil inoculum. So it wasn't so much just the case that all the genotypes responded the same on all the soils. That wasn't the case at all. We can see now that these genotypes have a genotype response among the soils that is at least partially unique to that genotype. And so where does this lead me? That's me washing plant roots in the lab, probably very late at night. And so this leads us to these more questions. Obviously, this study is just, it's the starting point of the map that leads to my many areas of PhD research. And so we have these more questions. So do these genotypes assemble different root microbiomes? And so from all those pots that we saw in that greenhouse, I saved the roots and rinsed them. And they're all labeled and in the -20 freezer in plant sciences. And so whenever I get back to the lab, one of my first things I'm going to be doing is pulling these microbiomes out of these roots to see if these genotypes did assemble root microbiomes, because how do we explain these differences in performance on the soil types? And I think one of the places we need to look is in the root microbiome. What are the starting soil communities? The third question, did we fully account for abiotic differences among the soil inoculums? Because we weren't expecting the trends to be that obvious with this experiment. We could really see different, even just in the greenhouse visually, we could really see differences among the genotypes depending on the soil inoculum they were growing on. And so moving forward, we really want to make sure that our nutrient and our buffer solution are really accounting for any of those abiotic differences among the soil inoculums, because we really just want to be focusing on what role differences in soil community is playing on differences in plant fitness and the resulting plant community composition, which is that fourth point. So what effect would soil community have when genotypes are grown in competition for multiple generations? And this is really where we start getting back to the ecology level, and that now that we know that these genotypes seem to have, I say preferences, I don't mean to like anthropomorphize like these plants, but they have preferences for what soil they show better fitness on. And these preferences aren't the same across the genotypes. Some genotypes do better on certain soils. And knowing that, if we grew these plants in competition, would we see that certain genotypes performed better on certain soil inoculums reliably? So how would, if we mix the genotypes up and grow each of them on each of these soil inoculums, after 3 or 4 generations, at the end, would we see different plant communities structured by the microbiomes in these soil inoculums that we add? And this multi-generational experiment is really where this research leads. I want to say a big thank you to everybody, to my supervisor, Lindsay Turnbull, also Philip Poole in the Rhizosphere Lab. Andre is a postdoc. I harassed that poor man. Thank you so much. He's been so helpful. And also a big thanks to my whole lab group. And you can also find me on Twitter at CHNorto. Thank you.
So thank you, Christian, for your talk. I'm gathering questions from both Zoom and YouTube. So if you're on YouTube, feel free to post your questions on the live chat. If you're on Zoom, you can choose to ask your questions in person, post it on the group chat, or send it to me privately. So I have one question from Zoom. So it's a question from a non-plant scientist. How is autoclaved soil different from the other 4 types of soils, generally and perhaps specifically in terms of nutrient composition?
So when you autoclave soil, our main intention with autoclaving was to simply knock down the biotic component of the soil. We're not intending that autoclaving the soil kills all of the microbes. That's not the case. But we wanted to have a soil that was going to have a lot less microbes than the fresh soils. And so the autoclave, I'm not sure if that's part of the question, but an autoclave is like a very big humid oven that we put the soil in and then cook it and in some ways sanitize it slightly, maybe more than slightly. I think autoclaving does cause a nutrient burst in the soil. That's something that we've discussed, and our intention is to correct that with the nutrient solution. But then I made that point in the where does this lead section that we really, really, really want to make sure that we're correcting for these abiotic differences moving forward with the research. And that's something that we're very aware of.
Thank you. So I'm just waiting for more questions. Okay, there's one from Zoom. How do you think this research could translate beyond Arabidopsis?
It's a great question. So the Arabidopsis is, it's the model that we're using. And so I guess the reason we use these models is because the real life system is often so complicated that, I mean, it would take, you know, like these in, is it in situ? Like these in situ ecological research projects, they take years upon years and years and years and years to notice any kind of concrete trend from simply the noise of the data. So using Arabidopsis allows us to answer these questions in kind of a more direct way. But then we have to be more careful in terms of how these questions generalize for as you scale them up. And so the point of this is just to show, is hopefully show that these soil communities have a role in affecting plant competition and plant community composition through influencing plant fitness. And so the point of this research is to inform what we know about the forces that structure a plant community. So we know what pH does. We know, like gradients as you move along the poles. But now in this, we're trying to specifically ask what role do microbes have in structuring these plant communities? And so it's applicable in a broader ecological sense, past just what it means for Arabidopsis.
Thank you. So, there's one more question on Zoom. Were those 10 different variants of Arabidopsis that you used naturally occurring, or were they genetically modified in the lab?
These are naturally occurring. So these are accessions. And so I believe accession means a plant collected from a specific area. Yeah, these were all Arabidopsis that were found and collected in the wild. And then I got the seeds from the Arabidopsis Stock Center in Nottingham. And so they maintain these collections of accessions from across Europe and I think North America. But yeah, these plants, the accessions I picked were mainly from Europe and a few from North America.
Thank you. So if there are any more questions, please post them on Zoom or YouTube. And I think Christian will be able to answer them after the second talk. So now I'll move on to the second talk, which will be delivered by Ella, who is a third-year plant scientist at Cambridge, third-year undergrad plant scientist. And she will be talking about the pyrenoid, almost an organelle within an organelle, which is a fascinating type of carbon concentrating mechanism, which research into it possibly helping to find ways to increase crop yields. This talk will discuss the research being done into this structure and how it could possibly be engineered into crops, as well as how interesting it is just by itself. Without further ado, I'll welcome Ella to deliver her talk.
Cool, thank you. I'll just share my screen. Okay, cool. Awesome. Play from current slide. Okay, cool. Is everything working technical wise? I apologize for the weird lighting in here. Cool. I'm going to assume everything's working. Okay, so thank you everyone for coming and sticking around. I can't promise I'm going to be as interesting as Christian, but I'll try. I also apologize for the mildly melodramatic title. I was expecting that everyone else was going to go with, like, you know, light-hearted titles rather than very serious ones. And I thought of this within 3 seconds when I was filling out the form because I had no idea what to write. So it isn't going to be anywhere near as dramatic as the title would suggest. But anyway, my name is Ella Catherall. I'm third year Bionasci at Cambridge, where I'm specialising in plant sciences this year. And as was mentioned, I will be talking about the pyrenoid. I first became interested in the pyrenoid when I was actually preparing a talk I gave a couple of months ago for Cambridge BioSoc about the value of chloroplast research and the implications of chloroplast research for helping to solve problems that humanity is facing. And I read a few papers on the pyrenoid. I'd heard of it vaguely before, but had never really explored it. I read these papers and was like, oh my God, this is like just the most incredible thing I have ever seen. And so, yeah, it's really great for me that I now get to do a full talk on it. So thank you very much for giving me this opportunity. But yeah, before I properly go into the pyrenoid, it's worth me talking about why it is that it needs to exist in the first place. The main reason for this is this enzyme here. This is Rubisco, and it's shit at its job, which I relate to on a personal level. Rubisco is, as most of you will probably know, is responsible for one of the reactions in the Calvin cycle, which is part of the light independent stage of photosynthesis. And it does this bit here. I'm assuming you can see my cursor. And in this, what it's responsible for is catalyzing this reaction between carbon dioxide and ribulose bisphosphate to form an unstable 6-carbon intermediate, which immediately breaks down, relatable again. So the products can then kind of keep going through the Calvin cycle and eventually kind of generating stuff which can then be converted into glucose. Rubisco has one job. It just needs to catalyze this one reaction. However, sometimes it decides that it wants to go rogue and will go, you know what, I'm bored of carbon dioxide, and will just randomly decide that it wants to catalyze a reaction with oxygen instead. Now, the issue with this for the plant as a whole is the reaction with oxygen produces this molecule called phosphoglycolate, which is toxic to the plant. So the plant then has to expend energy in dealing with the phosphoglycolate through a process called photorespiration. And the energy it uses for that could be put to much better use, such as growing. And from an agricultural perspective, this will lower crop yields if it's having to put all this energy into dealing with phosphoglycolate. If you could stop Rubisco from doing this ridiculous side reaction, it would theoretically increase crop yields. So that would be great given growing global population. So a lot of plant science research at the moment is based on trying to make Rubisco less crap at its job. So and one of the main ways that evolution has figured out how to sort of increase Rubisco's efficiency is through the use of these things called carbon concentrating mechanisms. The basis of carbon concentrating mechanisms is just kind of surrounding the Rubisco and carbon dioxide so it can't come in contact with any oxygen. And as a result, it can't do the side reaction it's not supposed to do. It's kind of like blocking websites on your browser when you're supposed to be doing work, that kind of thing. It's preventing Rubisco from getting distracted. And there are various flavors of carbon concentrating mechanism. There's C4 rice, C4 photosynthesis. There's currently a project being done to try and engineer C4 photosynthesis, or the ability to do C4 photosynthesis, into crop plants, which is called the C4 rice project. There's CAN photosynthesis, there's loads. But these are all kind of present in land plants. And I think something that's often overlooked is that algae have exactly the same problems as land plants when it comes to Rubisco just making problems for everyone involved. However, algae, if anything, they're starting from a worse place because algae live in aquatic environments. In this case, we'll be focusing on green algae rather than red algae, but both live in aquatic environments. And the issue with living in an aquatic environment is there's less light in the environment. So for a photosynthetic organism, that's automatically a little bit of a problem. Most light present is blue rather than it being the really nice red that photosynthesis needs. The other issue is that in sort of an aquatic environment, there's less carbon dioxide present in the water, and also it doesn't diffuse quite as well. Most of the inorganic carbon is present in the form of bicarbonate or hydrogen carbonate ions, etc., etc., etc. So basically, they are screwed in so many different ways. And so it makes fundamental sense that the algae have over time evolved a way to increase their efficiency of photosynthesis, to try and make sure they're just that little bit less screwed. And this carbon concentration mechanism is called the pyrenoid, which is what this is going to be about. It looks like this. This is an electron micrograph taken from one of my favorite ever papers, which shows you what the pyrenoid looks like. Now, in practice, this is taken, the pyrenoid itself is actually present within the chloroplast, within the algae. And I would just like to take a moment just to appreciate how cool that is. The chloroplast already has such an incredibly interesting evolutionary history. It's what got me into biology. And so it's already having to do all these really complicated interactions to allow it to function. However, you then kind of go, okay, right, so it's doing all these complicated things, and it's also able to form an organelle within itself. Like, just, I think that's really cool, and that's why I find the pyrenoid so interesting. But yes, this is kind of what it looks like in a chloroplast. And so what we're going to talk about now is kind of how it forms, how it works, and then whether there would be any possibility of being able to engineer this into crop plants. So the way it works, one of the other reasons, other than just general chloroplast interest on my part, that it's interesting is that the pyrenoid is an inducible carbon concentrating mechanism. Things like C4 photosynthesis, CAM photosynthesis, those are things that the plant are able to do whenever they like. It's just there. The pyrenoid can just kind of come and go pretty much as it pleases. It will tend to it'll form when there's really low carbon concentrations in the environment. It's an inducible system. And the way it assembles is dependent on this protein here, EPIC1, which has a great name. I don't know if it was named explicitly for it to be pronounced EPIC. I like to think that it was, but regardless, it's called EPIC1, and that is responsible for the assembly of the pyrenoid. And what it does is, so EPIC1 contains 4 repeats, and each of those repeats are able to interact with a different Rubisco molecule. So one EPIC1 molecule is able to interact with 4 Rubiscos. So what it does is it kind of connects them all together, and that then forms this wonderful lattice, kind of shown by this kind of diagram on the side. And just as a side note, which we're going to come back to, the EPIC1 is interacting with 2 alpha helices present in the small subunits in the Rubisco to form this kind of hexagonal arrangement. So what it's serving to do is collect all of your Rubisco together in one place. It's kind of returning to our original micrograph. That's what this middle section here is, this higher section. That's just all Rubisco that's being shoved together. Now, as I said, this is kind of, the pyrenoid is essentially an organelle within an organelle. So for it to be an organelle, you need to have something that's kind of delineating the contents of the organelle from the contents of the cell. In this case, this is this kind of starch sheath around the outside, which is made of starch plates. And then you also have to have ways of getting stuff in and out of an organelle. So in that case, it's these thylakoid membranes that differentiate to form kind of tubules that allow things to both enter and exit the pyrenoid. So how it actually works, you've assembled it, everything's going great. How does it actually work? Yeah, this is again taken from like my favorite paper. So as I said before, in an aquatic environment, most of the inorganic carbon is present in the form of hydrogen carbonate ions. So what happens is the hydrogen carbonate ions are actively transported into the thylakoid kind of tubules, and then kind of move along, and we'll eventually encounter a carbonic anhydrase enzyme, which is able to convert the hydrogen carbonate into carbon dioxide and water. And the carbon dioxide then kind of moves out over all of the Rubisco, and the Rubisco goes, yes, okay, I remember what I'm supposed to do now. I'm not doing that weird oxygen thing. I'm just going to do the stuff with the carbon dioxide. The products of that reaction are then made and are then able to be kind of shipped out through other thylakoid tubules to then kind of rejoin the Calvin cycle, and that can then keep going. I think it's quite an elegant system. I think it's kind of, it works very well, and I think it's really cool. So, and like the impact of this on the ability of the algae to sort of function cannot really be understated. And I think the effect it has is nicely shown by this experiment that was done by the Griffiths lab in Cambridge, where what they were looking at was pyrenoids in different green algal species, and also the enzymatic kinetic parameters of the Rubisco in those different species. So we have this kind of table of data, which I'll kind of quickly run through. So what you have on this kind of top row here is you have Chlamydomonas, which is the model organism for green algae. You can just ignore that row, that doesn't need to be there. Then all but the bottom row, those are the different green algal species they were looking at. And then this bottom row is Arabidopsis, which, as Christian mentioned, is the model organism for land plants, and so angiosperms. So, and also column-wise, we have KCAT and KC. I hate enzyme kinetics. I hate that I have to mention it, but it's a cool experiment. So KCAT is the turnover rate. So it's kind of measuring how fast your enzyme is able to work, basically. And KC is a measure, I guess, of affinity of the enzyme for its substrate. So when they were looking at the pyrenoids present in the different green algal species, what they found was that 2 of them weren't quite as good as the others at kind of having a functioning pyrenoid. It might be that they were kind of leaky, because different pyrenoids look different in different green algal species. What you'll often find is they'll have different thicknesses of starch sheath, or they might have different numbers entirely. So these 2 were the ones, so you have Klebsidium subtilis. I apologize for any Latin errors. I didn't go to private school, so I don't speak Latin, and Spirogyra. they had kind of pyrenoids that weren't quite so great. And if you look at the kind of catalytic parameters of their Rubisco, it's interesting because you'll see their K-cats, both of them are higher than for the others, like the other green algae. And the Kc, in the case of K subtial, is much lower. So the interesting thing here is saying, okay, their Rubisco is working faster, and in the case of K subtial, it has a higher affinity. So basically, if you have a good pyrenoid in the case of all the other ones, your Rubisco can kind of slack off a bit. It can be even more terrible at its job if you have a pyrenoid to make up for it. Even more interestingly, if you then compare that to the analogous parameters in Arabidopsis, you'll find that the values for these green algae with not quite so good pyrenoids are much closer to that of the land plant. Not having an amazingly great pyrenoid results in the evolution of the Rubisco shifting to kind of more closely follow the route that land plants have taken. Pyrenoids are shaping evolution here, which I think is really, really cool. So obviously, I hope I'm kind of selling to you the fact that pyrenoid is a really good thing that evolution came up with. It's great. So the question is now, can we put it into crop plants? Because if you could, it would kind of prevent Rubisco from doing this rogue thing that it does sometimes. Crop plants wouldn't need to be constantly putting out phosphoglycolate fires. They could put far more energy into growing, which would mean more food for us. And that is, more food is always great, but also particularly when you're trying to solve world hunger, particularly when you have a growing global population. There's a lot of things that humanity is going to have to deal with. Now, there are reasons for optimism with regards to this. So for those of you familiar with the C4 Rice project, you'll be aware that one of the reasons why the people behind that think that it should be plausible is that C4 photosynthesis is thought to have kind of evolved independently a number of different times. And that should possibly suggest that different crop plants should be at least open to the idea of taking on C4 photosynthesis. Similarly, the pyrenoid has a kind of similar evolutionary history. It's kind of been lost and gained a whole bunch of different times by different green algal species, also present in hornworts, which are a type of early land plant. What I think is even more interesting is that cyanobacteria have something that kind of look vaguely like a pyrenoid. It's not a pyrenoid, they're called carboxysomes. There's two different types, alpha and beta, depending as to the type of Rubisco included, and they assemble slightly differently. But the main thing is they're based on the same principle of we're just going to collect together all the Rubisco in one place and ship the carbon dioxide there, and which is basically kind of what a pyrenoid is doing. Even more interestingly, if you look at how they're assembling, you know how I mentioned before that EPIC1 interacts with the small subunits of Rubisco. It's kind of similar in this case. So the assembly of alpha carboxysomes is controlled by a protein that interacts with the small subunits of Rubisco. And in the beta carboxysome case, it's the protein itself, the repeats look like the small subunit. So clearly that is apparently the way to go when you're assembling your pyrenoid. However, there are a number of issues when it comes to trying to engineer this into higher plants, because surprise, surprise, it is complicated. Because this is biology and this is how biology works. Oh, hold on, click the wrong button. I apologize. But yeah, biology is complicated, as are computers apparently. And so this kind of is why it is that it's kind of difficult to engineer into crop plants. One of the main issues that we're running up against is a lot of the Rubisco that's present in crops isn't necessarily pyrenoid compatible. If you try and, it doesn't really like interacting with EPIC1. So you can't get it to form this nice hexagonal kind of matrix that you would want, that you see it form in green algae. Obviously, the way to maybe make that work would be to swap out the land plant kind of small subunit sequence for that from green algae. But obviously, that is going to introduce problems of its own. So again, quite complicated. The other thing is as I mentioned before, the pyrenoid is an inducible system. And so we still have a lot of questions about how it's regulated. And those, understanding the genetic regulation there is going to be really key to being able to engineer it into crop plants. And we just don't quite have that understanding yet. There are certain elements that seem to be regulated a lot by phosphorylation, which is great. But obviously we then need to identify what are the kinases, what are the phosphatases that are responsible for that. Thylakoids are also really confusing in this case because you have these thylakoids that just kind of penetrate in, that allow you to have stuff in. Why is it that thylakoid membranes suddenly decide to have a career change? What's regulating that? So that's one of the other questions. We have no idea what's going on there. Also, bicarbonate transporters aren't currently really present in crop plant chloroplasts or in the thylakoid membrane. So we need to find ways to express those. There has been some progress made with this where they've been expressed and they show up in the right place. The issue is they don't work yet because, again, we don't understand how they're regulated. And so there needs more research needs to be done there. The issue is also, even if you were to get your bicarbonate transporters to work, the carbonic anhydrase in a lot of land plant chloroplasts it likes doing its job, essentially. So if you had bicarbonate kind of around, the carbonic anhydrase would go, oh, great, cool, more substrate, and would immediately dehydrate it. So you wouldn't get your carbon dioxide concentrated around your pyrenoid as you would probably want. So yeah, there are a number of issues. But I think this is my references as I do my little final bit. But basically, the pyrenoid is really cool. I think it's definitely something that is worth looking into, the possibility of putting it into crop plants. But fundamentally, I think it's just a really, really interesting system. And I think it's something that we should look more into, both to kind of try and understand chloroplast evolution and, yeah, just generally, I think it's just fascinating that it exists. So thank you. Stop share.
Thank you, Ella, for the very fascinating talk. So I'm once again gathering questions from YouTube and Zoom. There's one question on Zoom. Are there any hypotheses regarding the regulation of thylakoids?
So not as far as I could find as to sort of how it is that those are regulated. That appears to be one of the main questions about how the pyrenoid actually forms at the moment. So not as far as I'm aware is my answer to that one. I apologize.
Thank you. So yeah, if anyone wants to ask questions in person, feel free to unmute or just let me know. So I myself actually have a question. I did not do plant sciences. So you mentioned that there were starch around pyrenoid. So what are the functions of those starch?
Yeah, absolutely. So the reason that starch is there is, so if you just had your Rubisco just kind of concentrated in a place without kind of separating it from the rest of the stroma, you'd get your carbon dioxide go in, but chances are it would probably diffuse out. You wouldn't be able to concentrate it quite as effectively. The other issue is you'd also get oxygen moving in. So what the starch plate is basically there to do is to keep the carbon dioxide in, because obviously it's able to enter in through the thylakoids, which the oxygen isn't able to do, and keep the oxygen out so that the Rubisco doesn't go rogue, basically. So yeah.
Maybe one follow-up question from myself. So do those starch, I assume they just come from the chloroplast as a product of photosynthesis, and do they, because you mentioned that the pyrenoids are inducible, so when I assume they get broken down after they're not needed, and are those starch molecules digested and used up by the plants?
So obviously, I'm not entirely sure with that. The main kind of, as well as the kind of questions about how the thylakoids differentiate, we also have questions about how the starch actually assembles around that, because that, as you've identified, is really weird. Why is it it's just forming these plates in this area? Just what? So yeah, there are a lot of questions about that. And then again, I said there are also then questions about how it is that then breaks down and how it reassembles. So yeah, unfortunately, we don't know is the main question, which is a lot of the answers to a lot of questions about how the pyrenoid actually forms, other than EPIC1, which we do seem to have a decent understanding of now.
Thank you. So there's one question from Zoom. Has land plant carbonic anhydrase been engineered to reduce its efficiency in any experiments?
So it might have been. I don't know of any. It seems like something that should be feasible. Of all of the kind of issues that I threw up, that seems to be the one that should be possibly easiest to mess around with. It's just obviously chloroplasts are in their own way, almost an organism within an organism. So it might be that kind of messing around with the activities of different enzymes would affect their functioning. So I don't know of any experiments off the top of my head. There might be some, but I think, yeah, you would possibly run up against issues with regards to chloroplast functioning, but of the issues, that does seem the one that would be kind of easiest to figure out, I guess, based on my understanding. So.
Thank you. So I guess there aren't any more questions. So I will pass the floor to Christoph to wrap up this session.
So once again, thank you, Christianella, for 2 brilliant talks, with the first one focusing more on perhaps microbiomes and soil communities and how they affect plants, which is a very important issue nowadays, especially as we're trying to work out how to improve efficiency, to find out more about how these interactions can affect our crop plants, how they can affect whole communities. And of course, thank you, Ella, for coming back to speak to BioSoc. Thank you for that wonderful talk, very engaging as always. And thank you for everyone who asked. Thank you to everyone who asked questions, who engaged with the talks. It was a pleasure hosting both of you, and I hope to see as many of you as possible tomorrow for our 3rd day of Varsity Ski, and we'll kick off at midday, I believe. So thank you everyone for joining us, and I'll see you tomorrow. Thank you everyone.